Good afternoon, everyone. So first, I would like to thank for the opportunity to present my lecture here today, in which I'm going to tell you the story of why we started using the Bromodeoxyuridine or BrdU Comet Assay, and what are its applications and the main differences from the standard Comet Assay. As a way of introduction, I'm a postdoc at the Department of Molecular Biology of Cancer in the Institute of Experimental Medicine of the Czech Academy of Sciences in Prague. And our department is headed by Dr. Pavel Vodička, and our main research interest lies in investigating molecular characteristics of solid cancer, mainly those related to DNA damage and repair. I was also a member of H-Comet Cost Action, and currently I'm involved in ICAO-G committee. So since we have been using the Comet Assay in our project for many years, I was contacted by my colleague from another institute of the Czech Academy of Sciences to perform some Comet Assay experiments for their project. So we performed several experiments on mouse thymocytes, and since we didn't see any differences in DNA damage in different population of cells, or on the other hand, we saw DNA damage in those cell population in which was no logical reason for the presence of DNA damage. We started wondering if the final amount of DNA damage could be affected by the fact that the cells may be at the different stages of the cell cycle and may contain also replicating cells. So we started searching in the literature and for the possible improvement of the Comet Assay, and we found the option of using BrdU labeling in combination with standard alkaline Comet Assay, as published by two research groups between 1999 and 2013. So what is BrdU? BrdU, or 5-bromo-2-deoxyuridine, is an analog of the DNA precursor thymidine, and is incorporated into newly synthesized DNA by cells entering and progressing through the S phase, or DNA synthesis, of the cell cycle. Incorporation of BrdU has been established as a popular assay for determining cell proliferation rates in a wide variety of organisms. And BrdU Comet Assay is then the combination of immunofluorescent staining of incorporated BrdU and the standard alkaline Comet Assay analysis. The incorporation of BrdU can be revealed then in two-step staining, which includes incubation with primary anti-BrdU antibody conjugated with biotin, and in the second step you incubate it, or streptavidin CI3 conjugate is used for fluorescent labeling of DNA incorporated BrdU. So what are the applications of BrdU Comet Assay? This approach can be used for measurement of DNA damage in those cell populations that are unsynchronized, so in different phases of the cell cycle. Since cells progressing S phase form comet tails in alkaline Comet Assay, this approach helps to distinguish contribution of replicating cells to the total number of cells that form comet tails. Or in other words, it allows discrimination of the cells with DNA damage from those cells in the S phase of the cell cycle, which also contain a physiological level of DNA damage, either strand breaks produced during post-replication DNA repair or as gaps produced during semi-conservative replication. There is also one application, and it's pulse labeling of cells with BRDU. It has been used to test post-replication recovery after DNA damage, where cells with compromised post-replication repair machinery show a marked increase in the amount of BRDU labeled DNA in comet tail. So what are the differences from the standard alkaline comet assay? The protocol differs in two stages, and these are the preparation of cell suspensions, then the embedding of cells in agarose and cell lysis, and the comet visualization and analysis. So let's start with the differences in the preparation of cell suspensions. So a wide variety of human or mouse cell cultures or another cell population can be used for in vitro labeling of cells with BRDU. And in this step, the cell culture medium is removed and replaced by a fresh cell culture medium containing BRDU, and then incubated for a desired length of time at 37 degrees. In case of in vivo labeling of mouse thymocytes with BRDU, the BRDU solution is injected intraperitoneally into mice, and after 2.5 hours of incubation, the mice are sacrificed and the thymus is homogenized to produce the cell suspension. In the step of embedding cells in agarose and cell lysis, there is only a slight difference, and it is that after lysis, the washing, there is a washing slides 3 times for 5 minutes in cold BBS. The most different step is the comet visualization and analysis. So as I already told, after electrophoresis and neutralization, you have to incubate your slides with primary antibody, then with the secondary antibody, and then the slides are stained with fluorescent dye. How is it visible in the microscope? So the visualization of the standard comets and BRDU positive comets are visualized using 2 different filters. So one fluorescent filter is used for visualization of comets stained with YOYO1 for detection of real DNA damage, as you can see on the pictures on the left. And in the second filter, you visualize the BRDU positive comets as is seen on the pictures on the right. So the white circles show the BRDU negative comets. So then after filtering out the BRDU positive comets, it will ensure that that the qualification of the real, that you quantify the real induced DNA damage and not the DNA damage in combination with the physiological DNA damage, which may then false positively increase overall DNA damage levels in your experiment. So at the end, I would like to acknowledge all my colleagues who contributed to establishment or to this essay, and also to ICAOG, and thank you for your attention.
Do you plan to make to continue these validation studies comparing maybe traditional comet assay and in the sensitivity to evaluate the DNA damage in some known DNA damaging agents?
To be honest, not now, but yeah, it will be definitely interesting. So I hope I will find any students who want volunteer to continue with this. Yeah.
In the meantime, there's two more questions in the chat. First one is, would pulse treatment with BrdU impact the rate of the ribonucleotide incorporation into the double-strand DNA molecule? If so, could this increase the observed DNA damage?
Yeah, so we didn't use the pulse BrdU incorporation, but yeah, you can observe with this the differences in the maturation of DNA. So, and is it also possible to, yeah, yeah, if it could increase observed DNA damage, it could probably, yeah, it could increase, but to be honest, I don't know. I didn't try this, but yeah.
And then there's a question from Blanca. Do you have any example of studies obtaining different results in proliferating versus non-proliferating cells?
No, there are no studies.
Yeah, it would be interesting.
Yeah. So as I thought, only through 3 papers, and we were tested it, and no more studies.
So Sonia, when you did you do any analysis of the tail or percentage tail DNA between BrdU positive cells and BrdU negative cells? And if yes, did you see a difference?
Yeah, there was slight difference, but yeah, there was, but it was mainly because we were optimized for a long time the staining of the comets, because we tried using ethidium bromide and cyber green, cyber gold, and finally we used the yo-yo one as I presented, and so the difficulties were that the signal from BrdU sometimes appeared in yeah, go to the another filter. So usually in the BrdU comets have less DNA damage in tail, but it correlates yeah, the proportion you know, the few DNA damage in the tail correlate with the DNA damage. So there is less DNA damage in BrdU Comet Assay, but it is yeah, there is a correlation. So it's probably due to the method, but not due to the increase of DNA damage. If you understand.